Good afternoon, everyone. Welcome to our first webinar of the year of BePA, the Belgian Proteomics Association, for those who don't know what the abbreviation stands for. Today we present Tine. Tine is a fourth year PhD student for the moment, and she started off with a tissue predictor development in which actually she needed some metadata, and there she got to the PRIDE, of course, where the repository should have a lot of metadata. To her frustration, that wasn't the case. So she's here to convince you to put some metadata in there, and also the real data, of course. That's what most of us want to have in there. But definitely it's not ending there, like Tina will show you. Being here, I also want to take the opportunity to present some upcoming events of BePA in the near future. So we don't stop here with the first webinar. We will have a second one in May, June somewhere. So we keep you posted through the social media. So keep an eye on it. We also have the end of August, our BePA at VIB Proteomics Summer School. So definitely all PhD students, take a look at the website, at our social media as well, because registration will open somewhere next week for this. There will also be a Brooker event hosted by BePA in Ghent in the 7th of May. So might be interesting if you are interested in running some timsTOF experiments. And there will be a BePA conference by the end of the year. So also we don't have a specific date yet, but keep an eye on all our websites and social media, just in case you want to know some more about the BePA and our what are we doing in Belgium and outside. So now I'll give the floor to Tine. Enough about the BePA itself, and I'll give you the phone to Tine to give you a bit more information about PRIDE. Go ahead, Tine.
All right. Hi, everyone. And thank you, Anne and Simon, for the kind introduction and, of course, the invitation to come on here and talk about PRIDE in general. So why me? I think that's the first question to ask. Why am I here talking about PRIDE? Anne mentioned a bit on it. So I started out my PhD project in the group of Leonard Martens, Compomics. And what we do on a regular basis is actually just taking a bunch of the data from PRIDE, downloading it, reprocessing it, and then applying machine learning and deep learning on it to extract biological knowledge. In my case, that was tissue-specific proteomics. Well, because I'm reprocessing a lot of public data, my main promoter, Leonard Martens, he started the PRIDE database, and my co-promoter is Juan Antonio Visaino, who is currently heading PRIDE. So I work a lot with the people over there. We collaborate on a lot of these tissue-specific efforts. And of course, I have been intrigued by the lack of metadata in the repository. So that made me like super involved with PRIDE people, and I think that's why I'm here. So I'll first talk you through a bit about data submission in general, why it's good scientific practice, and the Proteome Exchange and PRIDE Consortium. Then I'll talk about the PRIDE submission pipeline. That's probably why the majority of people are here, to really know how to submit your data to PRIDE. Then I'll also talk a bit about some tools that might help you with it, that are not immediately connected to PRIDE, but are like a nice to have. And of course, how PRIDE could work for you instead of you only submitting to it. So this is the overview of a standard mass spec experiment, right? You have your sample, you do your experimental study on it, you throw it on the mass spec, you get the results, go to a search engine with it, and okay, just the Q&A pop-up. So you have the search engine identifications, then you do statistical analysis, and you write your manuscript. To have your manuscript published, you need to have your data submitted to a repository such as PRIDE. But why is this actually? So data submission is often seen as good scientific practice to make all data fair. And with fair, we mean findable, accessible, interoperable, and reusable. And this is a thing that has been seen already a lot in genomics and in transcriptomics, but now it's emerging more and more into proteomics. And there are two main reasons for this, not only because the good of our hearts to make all data fair, but also because more and more journals require this. For example, all the Nature journals, the Molecular and Cellular Proteomics, Journal of Proteome Research, etc., they ask more and more to have the data publicly available. The same thing is happening to funding agencies. They want to see the maximal potential of the money they give you. So if you put your data then publicly and accessible online, other people can use it and you can create more downstream value from your data. So this is only increasing. And you have several options where you can submit your data. So this is the Proteome Exchange Consortium. And this is a consortium that merges all public proteomics databases together and make sure that we're all on the same wavelength about what data do we store and how do we store it, etc. And as you can see, they are spread all over the world. And the main difference is in how they store the data. Some of them reprocess it, some of them don't. For example, Panorama is also more for the targeted side of proteomics. But the biggest one is PRIDE. And PRIDE is here. It's located in the UK, in Hingston, which is like really close to Cambridge. And it's connected to the European Bioinformatics Institute. And this is a figure I stole from Juan Antonio. And it shows just the increase in data sets that have been submitted to PRIDE throughout the years. And like this is an exponential curve. And last week, there was a tweet from Juan celebrating that they now have officially reached over 50,000 data sets present in PRIDE. So the data that is available in that repository, it's incredible. It's a huge amount of proteomics data. And if you then look, for example, which organisms are most present, not very surprisingly, human is the biggest one. And then we already have some model organisms such as mouse, yeast, Arabidopsis, etc. They're also annotated with diseases. So you can see that the majority of the interest goes to breast cancer disease and then colon cancer. And I just think it's really cool that you can have an immediate reflection of the kind of research that people are doing in these repositories. For example, if a new instrument is released and you can see it reflected in the type of data that's being submitted. Because the cool part about PRIDE is it does more than just storing data. Like there have been tools developed based on the reprocessed data. For example, in Compomics, we developed MS2PIP and DeepLC. So both of these tools took a lot of public reprocessed data and use it to predict molecular behaviors, such as fragmentation, spectra, and then retention time, and then combine it into MS2 rescore. And these are all things that would not have been possible without everyone submitting their data to these repositories. And they kind of have revolutionized the field. The same thing for knowledge bases, such as Proteomics DB and then the Expression Atlas, even now the single cell Expression Atlas. They combine all this data into just a comprehensive overview of proteomics. And it gives you a new way and like an eagle view on the data. But to get everything there and to have these downstream applications of it, you need to get it into PRIDE first, right? So how does this happen? This is the PRIDE submission pipeline. So how it starts, you as a scientist, you submit your data to PRIDE through the PX submission tool. You add data such as protein, peptides, identifications, quantifications, your raw files, and then it goes to the curators. So it is still a manual check. These people check if your data is correct, if there are files that maybe malfunction for one reason or another, and if everything is like clear, your data is submitted to PRIDE and you get an accession number. And these accession numbers always start with PXD, and they make it easy to refer to your data set. This is also what you will need to note down in your manuscript. Dataset is publicly available through PXD and then your number. So how do we start with it? This is here, the PX submission tool. The PX submission tool is a desktop application, so no need for command line, coding, whatever. You just go to the site that you can see at the bottom of the slide and you download it. You might need to install Java as well to get it up and running. But once it's up and running, they will require you to make an account, and then you get to the first step. And the first step is actually choosing between, do you want to do a complete or a partial submission? Now, the difference between the two lies in how you provide the results from your search engine. If you provide them using a standardized identification results format, such as MZ-IDEntiML or MZ-Tab, they will see it as a complete submission. Because a standardized format allows Pride to integrate your findings into the Pride framework and make them visually accessible to other people. Now, if you have a search engine that does not support MZ-IDEntiML or MZ-Tab, you can add them to a submission in the whatever format your search engine provides you, but then you have to do a partial submission. What is the effect for you? If you have a complete submission, your dataset gets assigned not only a PXD identifier, but also a digital object identifier. This makes it easy for people to cite your dataset, similar to how they would cite your manuscripts. So it does add a bit of extra value. Maybe we should first have an overview of the kinds of data you need to gather before starting this whole thing up, because it can be a bit confusing. So what you see here at the top is raw. The raw files, these are the direct output of your mass spec. This can be, the extension can be raw, with BAF, whatever kind of instrument you have. But this is mandatory information. Obviously, you can't submit to Pride's proteomics database without the real spectrum. The second thing are your results files. So these are your standardized identification files, meaning standardized, so in MZ-Tab or in MZ-IDEntiML. And these are mandatory for a complete submission. If you have them in a non-standardized format, they are called search files. They can be in whatever. If you use MassQuant, MaxQuant, there will be very different extensions to it. And these are only mandatory for a partial submission. And then you have your peak lists. These are your MGF, MZML files. And these are only mandatory if you do an MZ-IDEntiML submission. But they are also recommended to add for any other kind of submission you do, because it's just nice to have the MGFs present. I kind of ignored this column on the side. This is the matching thing that you need to do. So what happens is you have all these different kinds of data sets, data files, that still belong to one sample. So during the submission protocol, you will need to be able to match, for example, your raw files to your search or your results files. If you have an MZ-IDEntiML file, you will need to be able to match it to your peak files. So just that it doesn't get messed up and you start matching your results file to another raw file, because then it would be very confusing for other people. Now, these are not the only things you can submit to Pride. I added some suggestions, not everything on here. But the first one being the FASTA database. It's very recommended to add it, especially if you do experiments that have not a standard FASTA database. Think of, you have a very particular species that you're studying. You're doing proteogenomics, for example, other things that could require a different FASTA database than usual. It's nice to add it, because then other people can use it if they ever want to use your data as well. The second thing is the spectral library. More and more spectral libraries are being used in proteomics, but there are some differences in how they can be created. And just adding your spectral library to your results ensures the reproducibility of your results, which is always very recommended to do. And then you should have seen it coming at one point. This is metadata in SDRF format. It's also very recommended to add this. I will talk about how to get to an SDRF format of your metadata later on the webinar. And then the last one is the quantification output files. It's also a nice to have. It's recommended. It's good to add it if you want to. Oh yeah, there's a box just to emphasize the metadata. But okay, you have started your PX submission tool. You looked at the data files that you have available and you was like, okay, I could do a complete or a partial submission. Then the second step is just filling in the details of the project you're working on. Like, what is the title of your project? Which keywords can you associate with it? Then a project description, how did you process your sample and your data, and an experiment type. This is all very similar to what you're writing in your manuscripts. This is like the material and methods section that you have. But also keep in mind that this is what users will see when they go to Pride type in the PXD that is associated with your project, this is what they will see first. So it is important to be clear and concise in here so people immediately get an idea of what is in your data. After filling in the details, you come to adding the files. So you just click here, you go to your local storage and you upload the files. And then you assign them with a file type. And these are just like the capital letters that were in the little overview I provided earlier. So your search files, your peaks, this is like your MGFs. Here you can see that they have results filed in MZID extension, meaning that they are doing a complete submission. And they also added their quants, their FASTA, and like a spectral library. So this is like a very comprehensive submission. After this, you need to do the relationship between your files. This is what I mentioned earlier by like matching your raw files to the results files that you have, just to keep everything of the same sample together. This is what they're doing here. So they have their result file, their MZID, and then they added the relationship to the search output that they had here, then the peak, so the MGF file, and then the raw mass spec file. Now you have to do this for every file that you have. So this can be a bit time consuming, but it is very important to do this correct. So otherwise you will get complaints from the team of Bright. After this, you'll have to annotate your data. So this is just experimental details where you fill in your sample species, tissue, instrument, cell type, disease, even quantification method, and then experimental factor. What they mean with experimental factor is the thing that you studied. For example, here they studied the technical replicates. So they wanted to see if there were differences between the technical replicates, but it could also be disease status. For example, you looked at disease versus controlled samples. This is really what is driving your research. Why are you doing this? In the future, we hope that when people upload a metadata in SDRF format, they can skip this step. It's currently not possible, but most likely will be in the future. So you have your files, you have made the relationships, you added the metadata. Then the last thing to do is, where is this data coming from? Who is responsible for this? Who should they email when they have questions, actually? And this is your PI, the head of your lab. So you fill in the name, the email, and then of course, any affiliations. Also, if you are connected to, for example, the Human Proteome Project of the CPTAC Consortium, you can add it here. Or if you did reanalysis of other projects, it's also important to mention this here so they can connect the dots accordingly. And then you actually filled in everything. So this is the submission summary that you get, and you can see like all the files and their relationships are visible here. You can export it to just keep it in case. And then it gets sent to the curators of Pride. So you will get a submission reference. This is a reference number that will be used by the people of Pride to reach out to you if there would be something wrong. It's not a PXD accession number. So you cannot use this number in your manuscript. So this now will go to manual curators, and you have seen how the exponential increases in data sets in Pride. So it could take some time before these people reach out to you and say, okay, everything is fine with the data, here's your PXD accession, and you can send out your manuscript, submit it to whatever journal you like. So do take this into account when you're like putting a time frame on your manuscript writing, that it can take some time to go from submission to having the PXD that is required by the journal. Now, this all looks very simple, but it can be more time consuming than you think because of it being research, things can go wrong. And there are some tools out there to help you with it. So for example, MZIdentiml is a file format that is required for complete submission, but it's not supported by every search engine. So there are search engines, I've listed some of them here. The list is definitely not complete, that output it. So if you're using one of those, you can just easily make it available through the settings that you output your results in MZIdentiml. If that is not the case and you do want to do the complete submission because you need like the digital object identifier to make your data set citable, you could use PSM utils. I will talk about that a bit later on. The same thing for SDRF. SDRF is the metadata format. stands for sample to data relationship format. And currently there are some tools such as FragPy, Peptide Shaker, and MS Amanda that support the technical part of an SERF output, meaning these search engines can automatically fill in for you what fragment mass tolerances you use, the trypsin, did you use trypsin or another cleavage agent, etc. I hope to add Sage and MaxQuant soon to that list, but we'll see how that goes. But you still need to make an SDRF for the biological data that you have, or you use another search engine that does not support the technical part of it, then you can use less SDRF. So these are two tools that I will talk about to hopefully make your life a bit easier. And the nice part is that they're all online, freely available. You don't need to install anything. They are totally clickable. No need for any command line knowledge of bioinformatics. You can though. PSM Utils is actually a Python package, but there's a browser interface. It's available through psmutils.streamlot.app. And what does the thing do? It has two parts, actually. So the first one is PSM file statistics, is what you can see here. And you can just throw in your PSM file. And then it will give you like a quick quality control check. It's actually really nice. So it can immediately say to you, like, this is the number of PSMs you have, the peptidoforms. It can give you the target decoy distribution. So it's nice before you submit your data to Bright, you just go to PSM Utils, throw it in there, look, okay, this looks fine. Or your PI asks for like a really short notice, a plot to see how clean your data is. PSM Utils can help you with that. But the second part, which I think is also very nice, is the PSM conversion. So they allow you to convert one PSM file type to another. So for example, you can input here PSM output from Sage and then convert it to MZID. So then you can use it to do complete submission, which is pretty cool. But also if you have someone else did an analysis on another search engine and you want to make it easy to compare the two results that you have, these things are really helpful. Then the second part was the SDRF metadata part. And if any one of you ever tried to make it, I emphasize try, it was developed by UBIC, which is the European Bioinformatics Consortium, and then HuPO-PSI, the HuPO Proteomics Standards Initiative. And if you ever try to make it, you have to dig through like the paper, the GitHub page, then find all the right ontology terms that match your sample, because everything has to be machine readable. Then you have to understand how the structure of the thing works and make it in Excel. You can do it in Excel. Like if you have a simple experiment, it's not that hard. But if you don't want to spend a lot of time on it, I just took everything and threw it into LessDRF. It's similar to PSM Utils, so it's online available through LessDRF Streamlit.app. And it's also completely browser-based, no install clickable thing. How does it work? This is your homepage, and it immediately starts with some steps. So this is what you're supposed to do. It will walk you through the whole thing of SDRF. And so the first thing it will ask you is, do you have some local metadata lying around? You probably do. So no need to reinvent the wheel, right? You just upload the local metadata that you already have, and LessDRF will check if it matches the ontology terms, because they want everything to be machine readable. If it's correct, then you will get a green mark. If it's not, it will say, okay, this does not match the ontology. The columns that cannot be filled using your local metadata will end up here in this to-do list. So you get a to-do list of these columns, and every time you fill them in, it gets like checked off. It's very satisfying. And then it will start asking you questions to build the structure of your file. For example, it will ask you, are there multiple cell types in your data? So if you put yes, it will say, okay, how many are there? And are there sometimes multiple cell types within one sample? Because that will change the structure of your SDRF file. You didn't have to figure out how it changes the structure. It will do it automatically for you. And then you can fill it in using the ontology that's incorporated in here. So you can do an autocomplete search function, or you just go tree-wise through the ontology. But there's no need to like browse to other ontologies and see which terms match closely to yours. Everything is in there. Then the last part is actually a call to you guys. If you are doing some specific type of proteomics, like metaproteomics or single cell proteomics or immunopeptidomics, a kind of experiment that we do not have a standardized format for yet. Please reach out to me if you have some ideas on how we can make this better for you. So, because we try to make it as easy as possible for you to annotate your data. All right. So you spend a lot of time gathering your data. You do the experiments, then there's the mass spec time, there's the analysis time. So also the time to submit the data to PRIDE, because don't underestimate it. It can take some time, especially if you have a lot of data files. So it would be nice to know how PRIDE can be beneficial to you. I think that's fair to ask. So this is how PRIDE looked this morning. This is like just the standard interface. And the expression atlas and the single cell expression atlas I added to this because they're also based from PRIDE. But if you click here onto resources, it will show you 4 different things. First off, PRIDE archive. This is where all the data sets are stored. Then you have PRIDE archive annotated. This is just the same as PRIDE archive, but only with the data sets that have comprehensive SDRF metadata annotation. Then you have the USI, that stands for the Universal Spectrum Identifiers. And then the last one is the spectral libraries. And I will go over all of them and just show them how you can use them in your research. So, for example, you did your study and you want to compare it to what someone else might have done, something similar or something in another species, and compare are the same proteins affected, for example. So you go to PRIDE archive and you start here in the search bar with putting in some keywords. For example, you did a study on stress. You put those keywords in here, and then you here have some extra query fields you can use to filter down. These are based on the user-delivered metadata. And then you will end up with, in this case, 984 projects that studied stress in humans. You select one of those because it looks pretty interesting. And what you first get is here the summary. This is what you filled in the project description from the PRIDE submission tool, where it said, this is the first thing people will see when they look up your data. This is the example. So you can immediately check, okay, here they did 10 technical replicate measurements of a standard sample, for example. But you also have here on the right-hand side, the metadata that was inputted through the tool. So you can immediately see, okay, it was human sample, they studied lung. Oh, but it's lung adenocarcinoma. That might be not good for the thing I want to compare it to, for example. If you then scroll down a bit further, you have an overview of all the files that are associated to this project and a download option through FTP. And if there was an SDRF associated with the project, it's displayed here as well. This makes it really easy to reprocess another project and see how it compares to yours, because you know for every raw file, if it was, for example, here related to a 58-year-old, for someone else, it might be like 63. All right. Now, imagine you don't want to compare your project to an entire project of someone else, but just see you were reading something about the specific peptide and you want to check the spectrum for that specific peptide. What you can do is use the USI, stands for the Universal Spectrum Identifier. What happens is you go to PRIDE archive, you go to resources, you click on the USI thingy, and then you get this. And you can just put in here a peptide sequence, and then it will tell you all the projects this peptide sequence was found in. You can select one of them. For example, here I selected the PXD00561, that it found this peptide in the file that's started with adult frontal cortex, etc. And then it says a scan number it was found in, so you can really go back to the project if you want to. But here we just visualize the spectrum. And you can check, okay, did the peptide that I find, is it correct? How do we see the spectra? Does this compare to the spectra that I found in my project with associated with the same peptide, etc.? So I think this is like a pretty cool feature of PRIDE. And then the last thing, that was the spectral library, right? So spectral libraries are still, they are booming. I mean, it's really nice to have spectral libraries, but the way that they are built can be very different between people, like which tools they use, etc. So what PRIDE did is they made spectral libraries based on consensus spectra. And you can access them here. And if you use one of these spectra, it's really nice because you can just write in your manuscript. I used, for example, the Musculus spectral library from Pride. I downloaded at this date and it will be traceable and reproducible. But I think it's pretty fair to state that if you want to use all of this, like for Pride, that metadata is crucial to all of it. If you put your data on Pride, metadata is essential for other people to be able to interpret it. And it's just a good way to make sure that your data becomes your most productive legacy. Because if you have comprehensive metadata with your project, what will happen is other people will reuse and reprocess your data. This might lead to collaborations that you did not see coming before, or maybe citations, because other people will have used it and have benefited from it, so they will cite you. And it will just improve the long-term impact of your data. And I think it's very, very possible that in the future we will look at high impact data sets, the same as we look at high impact publications right now. So besides explaining everything about Pride, etc., this is also like a call to really annotate your data because your data is more important than you even know. All right, with that, I want to thank my colleagues. This is the Compomics group, a lot of people, but if you have any questions, I think this is your time. Thank you very much.
Thank you, Tine, for this very elaborative and very useful presentation. I did write down some stuff myself.
It's nice to hear.
Yes, some questions. What about DIA data? Is there already some consensus on how to be able to do a complete submission or some data formats?
Not yet. They are still working on it, and it's Pride still allows DIA submissions, etc. But I don't think it's possible yet to do complete submission because MZ-IDEntiML is not yet like able to support DIA as far as I know.
But it is in the pipeline.
Yeah, they are definitely working on it because more and more DIA data is being generated. So
Yes, and like the spectral libraries, do you have any idea if they use this already a lot for DIA, or rather they do it for DDA?
like they make DIA spectral libraries, you mean?
Yes, yeah, from the
Yeah, I
spectral
think
library.
so.
Yeah, that's nice.
Yeah, it is.
Um, so if there are any other questions, go ahead. Go ahead and pop them into the screen. I don't see anything coming. Well, you have everything from Tina. Oh, there is a question. Um, I'll just read it to you. I really back you up on the importance of metadata with tools to streamline metadata generation like LessDRF. Do you think Pride should make metadata submission mandatory?
Oof. Um, Personally, I think yes, but I do know, like, from my perspective, yes, but I do know from the perspective of Pride that it's not that easy to first convince everyone to do metadata annotation because it's not easy to do, even with less SDRF. It does require a significant amount of time. But I do think that in the future it will become that more and more important that I hope it evolves to being mandatory, but I don't think it will become that in the near future because not everyone is currently aware enough of how important it is and how to do it in the first case. But
Thank you. Any other questions or burning questions for Tina? Yes, there is. How does Pride handle targeted proteomics datasets? I might have missed that at the start.
yes, targeted proteomics, there are a lot of different repositories, and targeted mostly ends up into the Panorama database. So this is like if you have in the beginning this overview of all the proteome exchange data sets, they all have their own specific sub niche. The reason that they exist, because if they were all the same, there would be only one, right? So here you can see, so this is Pride, this is Panorama that handles mostly the targeted stuff. And then the difference between massive iProx and JPOS mostly comes from the way that they reprocess data, if they do it or they don't. But yeah, targeted, I would go to Panorama.
Perfect. Yes, from my own experience, I can only say that Panorama works. And thank you from the person that asked the question, Darshan. Any other questions? Well, with that, I guess we can close up here. Thank you all for being here. And thank you from some attendees, Tine, for your presentation.
Yeah, thank you for having me.
Very welcome. We'll see you on the next webinar, I hope. So keep an eye on everything, and thank you for being here. See you later, and bye-bye.